This is Alex Schreiber.
This is Chris Learning.
And today we're going to talk about mutations. Mutations are a stable, heritable change in the genomic nucleotide sequence. They can be either single base changes, also known as point mutations, changes of several bases, larger mutations, deletions, inversions, duplications, and also be translocations. So here we have CAU, UUU, and GUU, which encode for histidine, phenylalanine, and valine. So the first mutation we're going to talk about is silent mutation. Silent mutations are part of the point mutations, and they're alterations of the base sequence that do not alter the amino acid sequence of the protein because of code degeneracy. So here we have uracil, which encodes the phenylalanine. So a silent mutation is when a base part is changed, as we do here. We change uracil to cytosine, and it doesn't cause a change in the amino acid sequence. So here we still have, even though we changed the U to C, we have histidine, phenylalanine, and valine. The next mutation we're going to talk about is also known as the missense mutation. It's a point mutation in which the base sequence is altered, and it results in the incorporation of a different amino acid in the protein. At the level of protein function, the effect may range from complete loss of activity to no change in activity. So here we have phenylalanine, which is encoded for by uracil, and cytosine. If we add an adenine, an A, in place of the cytosine, the phenylalanine changes into leucine. So it shows that the base sequence was altered, and then the amino acids were changed. This is an example of a missense mutation, which is an example of a point mutation. The next mutation we're going to talk about is a nonsense mutation. Nonsense mutation is a point mutation, and it involves an alteration of the base that produces a translation termination codon. This results in premature termination of protein synthesis, and at the location of the mutation within the protein will determine the extent of the change in function. So here we have uracil adenine, which encodes for leucine. If we change the middle uracil to a guanine, the leucine becomes a stop codon. So therefore, the valine would not become translated into the protein, but the histidine would still be translated. This is, again, is an example of a nonsense mutation, which is an example of a point mutation.